time-invariant model of phenotype dynamics in breast cancer cell populations. This is joint work with UC Berkeley and the Oregon Health and Science University. This is a brief outline: introduction, motivation, prior work, methodology, mathematical overview, biological findings, comparison to previous work, and future work. So I will touch on many of the themes that Jennifer just discussed about. So phenotypic heterogeneity, or cellular diversity on the phenotypic level, poses a major hurdle to effective treatment of certain cancers. So discovering strategies that regulate heterogeneity is a key priority. And deeper understanding of drug-induced cell population dynamics is needed to design therapeutic approaches that control phenotypic diversity. So here we present a linear time-invariant model to identify phenotype-specific behaviors, division, death, or switching, that drive heterogeneity. So Gupta et al. Developed a Markov model that estimated phenotype switching probabilities using 2 time points, not incorporating division, death, or noise. We model phenotype-specific division and death using several time points, and our approach minimizes normally distributed noise. Alejandro et al. Modeled phenotypic spatial evolution in 2 dimensions, and switching rates were determined directly from the data. So we apply systems theory and optimization to estimate our model parameters. Finally, Goldman et al. Built an Lti model to represent net proliferation and phenotype switching. However, cell division and death are distinct in our model, and our method provides an initial assessment of therapeutic efficacy. So we are interested in understanding the population dynamics of 3 phenotypes: keratin 14 high, vimentin high, and not, which is everything else. These phenotypes are from the triple-negative breast cancer cell line, and we are interested in analyzing the dynamics under 3 distinct conditions: the control, GSK, which is a MEK inhibitor, BES, which is a PI3 kinase mTOR inhibitor, and combo, which is the unequal ratio combination of the 2 previous ones. So for each condition, measurements were taken from 4 wells at 6 time points every 12 hours. And so what do we measure? We measure the quantity of each phenotype, the quantity of dead cells, and the quantity of S-phase cells. This raw data is then processed, and then the processed data goes into our linear time-invariant model. The output of the model are the cell division, death, and switching parameters estimated using constrained least squares. Then we do some model validation in which we compare predicted and measured phenotype counts from test wells. We also perform statistical analysis. So we compute the 95% confidence intervals to determine whether or not differences between the control and the therapeutic conditions are statistically significant. And then we conduct biological analysis, which then generates new hypotheses to fuel future experiments. So let's take a quick look at what our process data looks like. So here we have, on the left-hand side is the control, and the right-hand side is combo, and you can see that the trends are pretty different. And so the role of our model is to analyze why those trends are different in terms of switching, death, or cell division. Let's take a quick look at what our model is exactly. So this is a schematic. It's very simple. So why are we using this Lti model? Well, in fact, the Lti model is the natural choice. And that's because a limited amount of data is available. Prior knowledge of phenotype dynamics is limited. And simple models naturally facilitate rigorous analysis. So this is the standard Lti dynamics equation, where X is our state vector. Our state vector contains 4 elements. The first three are the phenotype quantities at time k, which are green, gray, and red, and the final element is the quantity of dead cells at time k. So what is A? So A is our dynamics matrix that's going to be estimated using our data. So A contains the cell division, death, and switching parameters that are estimated using constrained least squares optimization. So now we will go a little bit more into the math. So this is the specific equation for our work. So we just have a 4x4 matrix, and we want to determine what the parameters in those matrix are using our data. So this is our optimization program. What we do is we minimize the difference between the measurements. We determine our matrix A by solving this least squares problem. And we constrain our dynamics A to have under biologically motivated constraints. Okay, so what about uniqueness? So in fact, convex optimization theory says that if an optimization function is strictly convex, then the minimizer, or A star, is unique. And so the strict convexity is satisfied when the x pre times x pre t, those are two matrices. That matrix is positive definite, and this is true for our data. So in fact, that our models A are unique, given the data that we have. Okay, so now let's take a look at our biological analysis. So what we did is, for each therapeutic condition, we asked, what is the existing hypothesis? Are the observed trends and the existing hypothesis consistent? And what cellular behaviors may explain the observed trends according to the model? So for each model parameter, which the model parameters include division, death, and switching parameters, we compare values corresponding to the therapeutic and control conditions. So this is a detailed look at some of our analyses, but I'm just going to point out two interesting points. So biologists believe that BES reduces the prominence of basal phenotypes, and basal is keratin 14 high. And so yes, we do observe this in the trends, but the model says that this occurs primarily due to less switching from the red phenotype to the green phenotype and the not phenotype to the green phenotype in comparison to the control. So this is interesting because one may expect that if green reduces under BES, then perhaps BES is killing green. But actually, the model says this isn't true. The decreases, in fact, occur due to switching, according to the model. Here's another interesting point. So combo is hypothesized to increase cell death overall, and yes, this is observed. But however, the model says that yes, there's more death in red and not versus the control, but however, there's less death in green under the combo treatment compared to the control. So the model is able to identify behaviors that can't be observed directly from the data. So these are just a summary of the key conclusions. So phenotype switching may play an important role in cell population dynamics, and at this point, empirical observation of switching is a current research challenge. And surprising findings motivate further study, particularly the therapeutic effect or lack thereof on death. Further, one may analyze the eigenvalues and eigenvectors of the LTI models. These are the A matrices that are generated for each therapeutic condition, to design therapeutic strategies that reduce heterogeneity. And so, for example, 10 sequential doses of GSK produces a population of about 70% green, according to the model. Okay, so here we will compare our method to a stochastic method from Gupta et al. So Gupta et al., their goal is to compute a stochastic matrix A such that this matrix equality holds. And so A contains phenotype switching probabilities for n phenotypes, and X0 and Xn are matrices that contain the phenotype proportions in n wells at time 0 and n respectively, and X0 is the identity matrix in Gupta's method. So we actually tried this method out using, on our data, using X0 that was just non-singular. And so it actually didn't work. We didn't obtain a unique stochastic matrix for our data. So an interesting question is, what conditions guarantee the matrix A, to be stochastic and unique? Okay, so the conditions which guarantee A to be stochastic and unique according to the literature are: 1, your data matrix D, which is defined as your as Xn, which contains the phenotype proportions at time n, and X0 inverse, where X0 contains the phenotype proportions at the initial time. That matrix D needs to have no negative eigenvalues. Number 2, D must be irreducible and stochastic. And 3, A must be non-negative. So this is just an example of a stochastic irreducible matrix with negative eigenvalues. Okay, so number one implies uniqueness. So why is that? Well, RECOMB is a mathematician at Cambridge, and this is from his theorem that describes the concept of a principal logarithm. 2 and 3 ensure that A is stochastic. So why is that? This is an application of a corollary by RECOMB and Lin that describes a necessary condition for A to be stochastic. So the key idea here is that Gupta's method motivates our new, more general approach. So just to conclude with some future work, we will construct a simpler model that can be validated using live cell imaging techniques in current development. We will model phenotype switching dynamics using a discrete time Markov chain with birth and death to determine the statistics of the biological process. Further, we will conduct an experiment with 15 wells per condition. In the previous data, we just had 4 wells per condition, because more samples are expected to improve accuracy of the results. And finally, we will use the model to predict therapeutic sequences that regulate phenotypic heterogeneity when data from additional drugs is available. So thank you very much, and I'm happy to answer any questions. I think we have a couple of minutes, right, for questions? You guys have time for questions? Questions?
So very nice stuff. I'm actually curious on your drug selection. So we just heard that triple negative breast cancers don't have very good targets, but yet you're using very targeted therapies.
Yeah, no, that's a great point. So I am a modeler by training. I'm doing a PhD in electrical engineering. So my expertise is really in the how, if we have this data, how do we model it in the most appropriate way? So I can't answer that question as of now, but I can take your email address and then forward it over to our biologist and then respond that way, if that's okay. Okay. Aren't you guys interested about how I validated the model? Maybe? How I did statistical analysis? No, I think that's like an important point, right? Okay. Oh, yes, question. Great question. Okay. So, sorry, I had to include a backup slide. So basically what I did is, so we used a very simple objective function. It was just, least squares objective function, minimizing the error, assuming that it was Gaussian. So we actually tried out other objective functions, more complex objective functions, but these actually didn't obtain clear qualitative improvement over the simple least squares. So what we did is we only have data of 4 wells. So what we did is to make the process easy, we used 2 wells for training and 2 wells for testing. So we determined our model parameters using the 2 training wells, and then we tested how well our model did on the 2 test wells. So in the diagram, in the graphs you see here, this is just for green, we see that the dotted lines are actually the predictions, so what the model predicts for every test well, and the solid lines are the measurements from the process data. So you can see that the model actually does pretty well with monotonic trends, but it struggles a bit with sometimes the directions of the oscillatory behavior. So in order for us to capture the oscillatory behavior, we would really need more time points and really need to model the second order dynamics. So this was just more like a qualitative validation. Yes?
Follow-up on that. Do you think that maybe by doing a 2-2 split, your model is not training on enough data? Have you thought about that?
Yeah, no, that's a great point. Yeah, so I definitely, there's so many different ways you can do validation. So there's, leave one out validation, and then you can end up using all the data. There are just so many different ways. But this was our first, our first trial, and so that's why Tyler, who is he's our experimentalist, he's going to do an experiment that's in progress on 15 wells per condition. So having 15 wells means that we have a much larger data set for training and for testing. Yeah, but that's a great point.